second peninsula, which we're going to go visit, which where the seagrass bed has disappeared entirely within a year or two. So there's something going on in this area, but we don't really know. We're trying to figure some of those things out. We think it's something to do with the physical environment and the water temperature, the currents, and nutrient delivery, and all that kind of stuff. So over there, past, if you can see around our boat driver here, so that's Second Peninsula. We had a seagrass site there for quite some time. It was a nice site because you could park on the road and roll in, and it was one of these sites that are on the inside of bays. It was really warm and shallow, and the sediment was really silty and had lots of organic matter in it, so there's lots of sort of toxic chemicals probably for seagrass living there. So we had one site there, and that's one site that has disappeared entirely. Around the same time that we saw our bed in Crescent Beach also disappear. So something was going on, and we weren't entirely sure what it is. But we tend to study these contrasting sites where we have these beds on the inside of bays, so they're probably more highly stressed because the water temperature is higher, doesn't get flushed as well. There's often, the light penetration through the water columns often not very good because there's sediments and particles in the water. So we have sites in a number of places like that, and then we also have sites more off in the open ocean where the currents are stronger and the water is a bit cooler, and so we'll visit one of those sites on the outside today, and that's at Sacrifice Island. But this is our Mason's Island site that we have here. We've studied this site for a couple of years. It seems to be fairly stable at the moment, but we're quite interested in monitoring this site and working here and trying to understand what makes seagrass resilient and persistent under change, particularly because it's in the same kind of region where we've seen these losses.
Too many nutrients go in from bad septic systems or just sewage overflows that can lead to an increase in algae, which can overgrow the eelgrass and cause a problem. The quality of the water, how much light is available in the water, and how many nutrients are in the water is probably the most universal problem for eelgrass. There's other things that are definitely problems too, but if you don't have enough light and you're getting overgrown by algae, then the rest doesn't really matter because you can't survive that situation. But yeah, it's all sort of interlinked. We can't think about just the eelgrass in isolation. We have to think about all the species that it interacts with.
Water quality is often blamed for seagrass losses, so that's just, thanks to us developing the upland coastal area, so we generally end up degrading water quality in the nearshore area. Runoff, things like that, wastewater, but then also just turbidity, activities that we do in the water that create turbidity, boat moorings and boat traffic and dredging channels so that boats can go through, things like that. And then there's things that are somewhat out of our control, like rising sea temperatures and weather patterns, more intensifying weather patterns.
So green crabs are an invasive species. They came here back in the early 1800s, and they just expanded along the coast of North America. They're actually found pretty much all over the globe now, and they can tolerate a range of environmental conditions, and that's why they're able to expand their range fairly rapidly. So green crabs utilize eelgrass beds to forage for food, and sometimes when the populations become very abundant inside an eelgrass bed, they may also uproot the plants. And so up in Maine, back in 2012, they lost, I think it was about a couple of 100 hectares of eelgrass due to green crabs foraging inside the eelgrass bed. They have a hyperabundance of green crabs in Plum Island.
We look at seasonal variations. We also look at differences between sexes. So we try to see if our males more abundant in the spring, summer, or fall, or females, when they're the most abundant. And then we also look at differences in size classes. So do we normally trap smaller crabs in the spring compared to the summer or to the fall? So what we find is that females seem to be most abundant during the summertime, And so from a manager's perspective, they want that information because if you're going to implement a trapping program, you want to be trapping when you have a lot of females because they're the ones that are producing the eggs.
And one crab produces how many again?
160,000 eggs. So it's worthwhile to invest your time in the summer and in the early fall when you see all the females coming out. They're pretty aggressive. They're also cannibals, so they'll eat each other.
Well, we know that they're there, but we've also always seen them there. So while they've become more publicly known about and concerning, we have seen them for a long time. They're not a new species here. And there has been some evidence of places where they've impacted eelgrass beds and soft-shell clam flats. But at the sites that we've monitored, we haven't really seen them as a primary stressor. They're there and they might contribute to the overall stress that the meadow is experiencing. But like I was saying, I don't think they're a smoking gun to a whole bed being devastated for the beds that we've seen. But other people, I know Hillary Nichols up in Maine felt that a whole entire meadow was lost due to green crabs. So really depends on the location and honestly whether or not people can collect data on them.
You agree, Boston Harbor, we kind of view crabs as not just look at the individual species, but crabs as a total, so all the different species of crabs.
Including natives.
And including native species, rock crabs, Jonah crabs, spider crabs, and blue crabs. We notice their impact more for the beds that we visit our restoration sites. If we notice them burrowing under our discs, under our restored grass, under what Joel described as a planting unit, sometimes we see them burrowing. So that's more of our issue, or if they clip, sometimes we'll notice that the leaves have been clipped by the crabs, so that's more of our issue here in Massachusetts.
We don't see a lot of green crabs at the sites that we've been working in. I know that they seem to be a problem in some places and not others. They definitely like to be ornery, it appears, and rip up plants when they find them. But thankfully, so far, we have not had a big problem with green crabs in our experiments. I know that they have been a big problem when we're trying to restore eelgrass because when you plant the shoots for restoration, it takes them a while to get effectively rooted into the ground, and so if the green crabs come and tug on them, they can rip them out of the ground very easily, and that reduces the success of the restoration.
Seagrasses are really important for so many different things. They are really important for promoting biodiversity in the ocean because there are lots of animals, fish and invertebrates, crabs, lots of things like that live in seagrass that can't do as well if you don't have a lot of seagrass around. It's really important as a nursery habitat for several important fishery species and species that we don't fish. So a lot of things like cod, baby cod, like to live in eelgrass. It's a good place for them to hide from predators so they don't get eaten. Flounder love to hide in eelgrass. So there are a lot of different animals that use the eelgrass, and if you don't have the eelgrass, or if you have less eelgrass, you'll have less of those animals, and there are things that we like to eat, there are things that we like to have in the world. So seagrasses are really important for promoting biodiversity and a lot of important fishery species. They're also very good at stabilizing sediments, so the water is cleaner and clearer. When storms come through, for instance, the sediments don't get as stirred up, and so the sand and the mud stays on the bottom, and the water stays clear. So again, we can use the water, we can swim in it, we can fish on it. And they're also important for protecting coastlines. If you have a storm surge coming in from a hurricane or a nor'easter or any big storm, they can help slow down the waves and slow down the storm surge and absorb some of that energy before it hits the land, before it hits the seawall or somebody's home or infrastructure like power lines or anything like that along the coast that's vulnerable to big storms and waves like that.
It's a foundation species, so there's a lot of different organisms that utilize these beds. And it also uptakes CO2 and stores it. It's considered a carbon sink, and it helps to improve water quality as well as clarity, which is important. And it also helps to stabilize sediments and reduce wave energy. So that helps to protect the shoreline.
It's really important for biodiversity. We tend to see higher numbers of animals living in vegetated habitats such as seagrass compared to adjacent bare areas. We tend to see higher levels of production, so these habitats are really productive, and so that helps the surrounding ecosystem because it provides food sources and materials that are used and nutrients that are used by the surrounding ecosystem. They're also really important for our shoreline, so they tend to be important for buffering the effects of waves against shorelines, and they help filter nutrients and contaminants from the water column. So there was a recent study that showed that they help filter bacteria. They form these vast underwater, beautiful, expansive meadows, but it's really, most people can't see that, from the road. So often we have these really valuable habitats present, but it's really, if we are from here, people don't often know about these habitats or understand how important they are.
So some of Jill's work is also just looking at more monitoring, the need for more monitoring of where we know eelgrass is or historically has been, just so we can keep track of it, try to pinpoint things going forward, so we don't know, like climate change, acidification of the water, how all of that together is going to impact
Yes, this is 9.
any of our beds.
And we have a whole other team of folks in the New Bedford office, and together with them, we've been trying to really bolster our acoustic mapping program. So we go out with side scan acoustics and map beds of interest.
Cool.
And every year we've been adding more and more beds onto the list of areas that we go to.
And
It's been really eye-opening too. So we do the side scan mapping, which is pretty high resolution, and then we compare that to the statewide mapping, which is done by Mass DEP. And they do an aerial photo survey, which is totally different, a whole different process. Seagrass net sampling is dissecting the plants that we collect. So during the dive, one diver is collecting a core. So it's a small core that you push down into the sediment and try to capture 1 to 2 plants, pull it up, and then we analyze it at the surface, figuring out how many individual plants are within that core, and then dissecting the plants into their different components to try to get a sense of the below ground biomass or living matter and the above ground biomass. And that's one of those parameters that you can track from year to year to see how the bed is responding to stressors and weather and things like that. Biomass is any of these, the pieces of plant living material. So in an entire plant, and I can show you a sample of that in a second, it's divided into the roots and rhizome, which are these components, and into the meristem, which attaches to the roots and rhizome and goes up to the leaves, and then the leaves. So we break it into these 3 components to see if there's any changes, track any changes across the seasons or across years. Wasting disease is not a new thing, but our ability to detect it has improved a lot. So now with Forest Help, we're using new methods to monitor and track wasting disease. Now we're scanning, we're creating scanned images of the leaves, and anywhere that there's black markings on the leaves. You can actually see it on some of these if you like. These black markings are wasting disease.
Oh yeah, I can tell.
So we're scanning them, and then they can be digitally analyzed.
So you can see the difference, this middle figure here is showing the difference between the summer and fall, or July and October, in the severity of infection, so the percent cover of disease on eelgrass leaves. And you can see that, it's hard to tell on this slide, but the higher infections are to the left and lower infection severity is to the right. And you can see that the October points fall more towards the right or towards the lower infection severity, whereas in the summertime or July, they're more towards higher infection.
okay.
But conversely, we see this figure here shows that the proportion of leaves within eelgrass shoot that are infected. We can see that in July, there's actually a lower proportion of leaves. So even though the infections are worse, there's a lower proportion of leaves that actually have infections.
And you
in
think
July.
that's because there's fresh new ones just
I think
move
that's
along?
because there are fresh new ones being produced so
Just quite
fast at this
fast,
time
right.
of year. And that's what's driving that pattern, yeah.
this is from the dissected plants. These are just the leaf part of the biomass assessment. So it goes in its own little bag here. And then these will all go into a drying oven for about 48 hours. Then we take them out and weigh the components individually. We get the dry weight. And then that's what we track over the years.
Here I have some samples that we've taken since Tay started the program in about 2008, 2007. So 4 times a year we go out to monitor this bed, and each time we take a sample back to do a plant press. So this is kind of our history of all the plants we've collected since we started. April 2017 is kind of up at the top. And that's kind of what one of the plants looks like in April. They're really short at our site. And right now we're processing plants in July. And typically you'll see a change in the size of the eelgrass throughout the year. And in the winter it's really short. The leaves are very, very short. And then by July in the summer they get very, very long. This is one in October, so just after. The leaves are a little bit more robust compared to the April plant. So they only grow for 2 years, and then they become a reproductive shoot and flower, and then they put seeds off, and then they'll die.
So when a bed is under a great deal of stress, sometimes the ratio of the biomass will change. So it'll start having a lot more above ground biomass, for example. The plants will try to grow taller to reach more sunlight. Seagrasses can grow intertidally and subtidally, but in Massachusetts it's really primarily just subtidal. So that means it's below the low tide line. Generally, it can only be accessed by scuba. There are a couple places in the state where if it's at the dead low tide, you can just access it by snorkel or by wading into the water. The majority of what we do, though, is subtidal, so it requires scuba, and also, in addition to the monitoring, we also do a lot of restoration. So we have a couple different planting sites in Boston Harbor and in Salem Sound.
Plants that came from different moms or from different areas and plant them together. We have found that they tend to have higher biomass. They tend to grow more quickly, and so and then from other experiments we found that if some sort of disturbance happens, then they tend to recover from that more quickly. And so that's something that we're looking at right now with regards to wasting disease, to see if that same pattern holds. So if you have a more diverse set of seedlings, are they better able to recover from wasting disease? And that's the experiment that we're still analyzing the data. It would definitely have implications for how we do restoration. So often we go to a single site and collect plants and transplant them in a new location. And so this might suggest that we want to collect from lots of different areas at that same site, or even multiple sites. So we're testing all of that now.
So you do see a little bit of fouling, but we don't see the really high fouling that you may have in other places. Without the high nutrient input, we don't tend to see that.
I study eelgrass and the amount of carbon that's stored in it. I've been working with the EPA for 4 years now, studying meadows up and down the coast of Massachusetts and as far north as Maine, and we've gotten down to Rhode Island as well. And we have some collaborators in other states and other regions, even some in Canada, and we're starting to compare our results across larger geographic gradients.
So 2 at 0, 2 at meter 3, 2 at meter 5?
I am marking these Ziploc bags, and the divers are going to take sediment samples, and they're going to take these bags underwater and put the sediment samples into the bags so that we can bring them back to the lab and measure the density of the sediments and the amount of carbon that's stored in them. Right now, we're just taking samples that are right on the surface, about 5 centimeters. We've cored this meadow before down to 30 centimeters with the big tubes. So we're not doing that this year because you end up just poking so many holes. We did it a few years in a row and found that the numbers were pretty consistent. So now we're just taking samples from the very surface, but we're taking them all across the meadow so we can see if there are differences between the amount of carbon that's stored at the edge of the meadow versus right in the middle of it, or if there's the density of the grasses makes a difference. So if you've got a really sort of sparse, patchy area, maybe that would store less. If you have a really dense area of meadow, maybe that'll have more carbon in it. So we're going to find that out.
I got a sediment sample for you.
Okay. Where is this from along the transect?
This is right at 0.
Right at 0.
So from now on we will.
Second 0 tag.
This will be for the second.
In the lab, we will be measuring the density of the sample and the amount of carbon that's in it. We measure carbon by, there are a few different ways to do it. For these, what we're going to be doing is you dry the sample, weigh it, and then you burn it. You combust it at very, very high temperatures. We put it in a furnace and we cook it at about 500 degrees centigrade. And all of the organic matter burns off and it leaves behind just minerals, things like silica and magnesium and things like that. And anything that's carbon or attached to carbon burns off. And so then we measure the weight of it after the carbon is all burned off, and we know that the difference was the organic matter. We have been measuring this meadow since 2014, and we started in 2014 with just, is there more carbon stored inside the meadow than outside the meadow? And we found that yes, there was. And that was just with a few sediment cores taken inside and a few sediment cores taken from outside the meadow. And we found a lot more carbon inside the meadow. So then the next year we looked at the middle, the depth range across the bed. And we had the question of, well, if we look at the sediments that are in very shallow water, where the eelgrass is very short, versus the sediments that are in very deep water, where the eelgrass is kind of long and flowing, versus in sort of the middle of the meadow, where the depth is ranges from about 1 meter depth to 6 meters depth. So it's a pretty big difference, and the eelgrass itself really changes in its shape and the way it grows over those different depths. So we found that there was the greatest amount of carbon storage in the middle depth, and a little less in the shallow, and a little less in the deep areas. This year what we're really interested in is small-scale differences within the meadow. The meadow is about 200 meters wide, so we want to know, okay, it's not just the shallow and the middle and the deep, but is this patch right here different from this patch that maybe only 5 meters away? And does it have to do with the density of the grass? So where the grass is really thick and growing very thickly, is it catching more carbon than the areas where the grass is a little bit more sparse and the coverage is not quite as great? Or does that not make a difference? So we're sampling every 5 meters across the bed today. We draw an imaginary line across the meadow, and then sample every 5 meters. We take a sediment sample and we count the density of the shoots of the eelgrass, and then we'll take a couple of samples of the eelgrass so we can measure it and see if it's tall, if it's wide, how many leaves each shoot has on it, so we get some idea of how the plants are growing in each of these different sampling areas. And then we'll measure the carbon in the sediments and put that all together and see if we have some patterns with the way the grass grows influencing the amount of carbon that's stored in the sediments. There are 2 things that we would do with this knowledge. One is probably convince people to do more eelgrass restoration so that we can promote the growth of eelgrass all around the coast here and hopefully all around the world. But even more importantly than restoration is protecting the meadows that we have already, so that the work that they're doing now isn't lost. The work that the grass is doing for us isn't lost. For the one for the eelgrass? Yeah, just like Nahan to the date. So, and the sediments will just be the same.
And so we collected seagrass plants in July, seagrass flowering shoots, and we put them in this tank out here in Hodgkins Cove in Gloucester. And back in July, we had 1,000 flowering shoots, green, beautiful shoots floating around here. But by now, they've been sitting here decomposing, and so you can see their biomass is all kind of rotting down here. And that's naturally what happens this time of year, and the seeds come out as the seed shoot decomposes. You can get the seeds out. So here's a seed. They look like little brown Tic Tacs. And so they're all falling out of the seed shoots. And then what we're going to do is take away the dead material and siphon out and filter the seeds that are lying on the bottom and collect them, and then we're going to plant them in October in Plum Island Sound and Salem Sound. We normally replant the adult shoots, and we have a method that we really like with a burlap disc where we tie 10 shoots into a burlap disc, and we put 5 discs in a meter squared area. And that plants the adult shoots out there. That's worked really well, but there are some reasons to think that maybe putting seeds in a certain site might do better. Seeds are negatively buoyant, so they drop right out of the shoots, and some of our sites that have certain hydrodynamic, certain circulation might be taking those adult plants away from the site, so the seeds aren't dropping out right there. So we figure at those sites where our plots aren't spreading through, it might be successful to actually put the seeds in. We've monitored the grass and the impacts of moorings at this site, and we've put in moorings that are low impact moorings out here in Hodgkins Cove to try to see if the grass will grow back if the mooring chain is no longer dragging.
We harvest from a variety of places, so it depends where we want to plant. So where we planted in Salem, traditionally we will harvest from sites that are nearby, since they're in similar depth and similar environmental conditions. But in the last 2 years, with the help of Northeastern, we've designed a study where we're looking at genetic diversity. So we're harvesting from 5 different beds, geographically separated from each other, all the way from Boston, inner Boston Harbor, all the way up to Niles Beach off of Gloucester. And we're testing to see if a mix of genetic diversity in the harvested plants will help create a more successful restored site.
I mean, there's definitely dramatic losses going on around the globe. I think we're also having some successes in some regions with restoration. Chesapeake Bay has had some success with restoration. We've had some successful restoration efforts here in Massachusetts, in Boston Harbor, and in Salem Sound. Division of Marine Fisheries has had several successful efforts where they've restored multiple acres of eelgrass. I guess I like to be an optimist and think more about how we can, what we can do than worrying about how much time we have.
So we selected a few donor sites. We did this so that we didn't have any serious impacts to one particular bed. We come in and we use our fingers to uproot the plants. We put them in coolers and we store them overnight. And then the following day we bring them to our location and we dig holes and put clumps of 5 into the holes and staple them down. It's really similar to gardening.
Limit their impact, of course, like if they're boaters, don't throw your anchor in eelgrass. Pump your boat out at a designated place, don't pump your waste into the water. But then if you live in the coastal area, reduce your fertilizer on your yard, things like that. If you want to get out in the field, try to find a local watershed group that does research or monitoring. There's a lot of those around here. There's actually a lot people can do to be engaged.
In general, people can be really aware of where they're anchoring their boats, so that they're not anchoring in eelgrass beds and ripping up the plants. We can be more conscious of water quality and what we're letting run off into the oceans, because that usually reduces light and can have a negative impact on the plants. And when you're applying your fertilizers, try not to apply them right before a giant rain event where it's just going to wash into the sewer system that runs straight into the ocean.
You can come out in the field and help us harvest and restore, but the best thing that everybody can do is try to reduce nutrient pollution, nitrogen into the system, so that's fertilizers and sewage runoff, because that is one of the biggest stressors to seagrasses and is causing a lot of declines in seagrasses around the world. And so, yeah, you can just reduce the amount of fertilizer you're putting on your lawns. That's would be amazing. Try to support upgrades in sewage treatment plants, and then also, as Fred always says, reduce the amount of of meat you're eating yourself for when there are issues with sewage coming into the water, because that would be less nitrogen. So become a vegetarian.
This was harvested yesterday. You can see the root, the leaves, these little nodes here where the root's coming out. We want to make sure that they're buried. Dig a little hole like that, maybe a couple inches deep. Pull the sand up. Then you want to put the staple over these right about this far down so that the roots are there. Go back to your hole, stick it in, hold them down, and then just cover back up with sand. Put them, maybe a foot apart, 6 inches apart.
Okay, so you see the seagrass. And my friend noticed that parrotfish were eating the epiphytes off of the seagrass. And she's actually, I think she finished writing her first paper, and she sent it in for publication. Haley Goss.
And they were made at the Alfred University School of Engineering, ceramic engineering. And so the plants can grow by spreading and creating rhizomes. And so they'll grow right off of this disc. We wove them in, and then we'll put them on a new location, and the plants will grow and spread, and we'll have restored an eelgrass bed that way.